further ado, I'm going to just introduce Dr. Christine Jones, who's coming to us from Australia. And actually, it's nearly 2 o'clock in the morning, I believe, Christine, your time. So thank you so much for staying up for us. Christine is talking today about the microbial life of plants, and she'll be taking a deep dive into it. And Christine was a keynote speaker at last year's BioFarm conference, and of course earlier this week. In fact, on Monday, Christine spoke. And she's worked for decades with innovative farmers, including the Danu Group in Ireland. And she'll probably want to tell you today, I think, that human life couldn't exist without microbes. In fact, if we want food security and to mitigate the effect of climate change, we need to work cooperatively with the microscopic creatures that, along with plants, compose 99% of all life on this planet. And us puny humans, not to forget, we comprise less than 1/100th of 1% of the Earth's biomass. And even within the human body, microorganisms outnumber human cells by 10 to 1. So, most importantly, I guess, Christine is probably likely to want to explain to us how we need to understand the crucial role that microbes play and how we need to work with, not against them. Because after all, every human civilization right across history that has degraded its soils has collapsed. And today, if we're not careful, we're on a similar trajectory. And as I say, a native of Australia, Christine has a distinguished record of implementing regenerative practices to enhance soils, biodiversity, nutrient cycling, water quality, and community and agricultural health. Delighted to introduce you, Christine.
Wow, thank you, John. That was a fantastic introduction. I appreciate that very much. That's probably the nicest introduction I've ever had. Thank you very much. And now that you've given my talk, we'll just go straight to Q&A. No, it's fine. I'm not covering any of the same material that you've mentioned, but it's a fantastic introduction to what I am covering, and I appreciate it very much.
Thank you.
So if I can get the technology to work, we're having a perfect night tonight. Everything is going to go perfectly today. I'm going to click on that and click on that and just do this. Slideshow, resume slideshow. That.
Yes.
Whoa. So what you can see there in that photograph is a plant root with lots of exudates coming out of it. Not because the plant wants to give away lots of energy, but because it's feeding the microbes. So they're the microbes that John mentioned in the introduction that we need to work cooperatively with in order to build soils and get nutrients into our plants and mitigate climate change. All of the above and a whole heap more that I'll be talking about today. So I'm going to speak fairly quickly and cover a lot of material in a short amount of time and leave some time for Q&A. Right, I could subtitle this talk, Root Mingling in the Soil Sociobiome, and they're the biggest words that I'm going to use today. I'll just leave that right there. I call it the sociobiome because there's a lot of interaction goes on between plants, and it's that interaction that is very, very important for all of the functions that we see that come from plant diversity, actually come from plants interacting with each other, or more correctly, they're microbes interacting with each other. So on Monday, I talked about the first payment that had ever been made in the world for soil carbon under a government regulated scheme. And when we see a significant increase in soil carbon when it's been measured to that degree, so that property was grid sampled to a depth of 1 metre across the entire landscape, that result is not anecdotal. We often hear people say it's not possible to build soil carbon, and any farmers who report that they've had an increase, it's just purely anecdotal. Well, we can't say that it's anecdotal anymore because it's not, it's real. And there has to be a lot of things happening in the soil microbiome in order for those increases in soil carbon to occur. They cannot increase at depth, in other words, below 30 centimetres, which is what I call sequestration of significance, the below 30 centimetre depth sequestration. With business as usual approach to farming, where we have monocultures, we have plants with very shallow roots, and we use high amounts of fertilisers and other chemicals, there is no way that we're going to be able to build soil carbon at depth, which is probably why the so-called experts will tell us that it's not possible. It's because where they're doing their measurements, it's business as usual, and it's definitely not happening there. So we need to make changes to the soil microbiome to enable that increase in soil carbon. And we have to have plants and microbes working together to produce that fertile, well-structured soil that's going to be high in carbon. So in John's introduction, he mentioned that 99% of the biomass of life on Earth is constituted with plants and microbes, and that humans actually don't even feature in that, because we're something like 0.01% or something of the biomass of life on Earth. So it's all about plants and microbes, and plants and microbes working together. And that's basically what I'm going to be talking about today. So what happens when they're working together? Well, on the left-hand side of the screen there, you can see some soil that doesn't have plants growing in it. And where this plant is growing is only like taken from, 10, 20 centimetres away. And you can see the huge difference that a plant makes to soil. And that's because of those root exudates that were in that first photograph and all the microbial life that is around those plant roots. So plants build soil, plants do not take from soil. If you have soil without plants, it ends up just being just minerals with no structure and no possibility for even feeding a plant, really. So plants and their associated microbes form a single unit, which is called a holobiont. So what is a holobiont? Well, a holobiont is a word that's occurring very frequently in the ecological literature these days. We see the words holobiont and holobiome and hologenome. So a holobiont is a host and its microbiome. So all living things are holobionts. So every living thing has associated microbes. So even microbes themselves are holobionts because they have other microbes that live within them and are associated with them. And together the host and its microbiome make the whole. And if we take away half of that, if we just have the host, which might be just a plant, and we take the microbiome away, the plant can't function because it needs to be a holobiont in order to function. So we are holobionts. So the human holobiont is made up of our genetic material and DNA that we inherit from our parents and our microbiome that we inherit from our mothers. And the importance of that human microbiome is becoming more and more obvious as we look deeper into human health and how our health is affected particularly by our gut microbiome. And, what was the microbiome that we did inherit from our mothers? And if it's deficient in something, is there some way that we can rectify that? So our genes determine, the phenotypic characteristics of us, our skin colour and our hair colour, etc. But it's our microbiome that has the largest influence on our health. And it's the same for plants. So our plant holobiont, the genes determine what species it is and what cultivar it is, but the microbiome determines the plant's health and productivity. So we need to learn more about the microbiome of plants. And the talk that's following mine today, which will be Professor James White, he will be talking in detail about endophytes, which are microbes that live within plant roots, and about how plants actually cultivate those and utilise those. And I think you'll find his talk quite fascinating because he has some amazing videos of microbes moving in and out of plant roots. And I'll leave it to him to talk about that. But how do we know, without having a microscope, how do we know whether a plant has a healthy microbiome or not? We need to carefully extract a plant from the soil and have a look and see whether it has lots of soil sticking to the roots. And if it has lots of soil, lots and lots of roots and finely branched roots, and Professor White will talk about the root hairs and root hair development and how that's stimulated by microbial activity. This is a very healthy microbiome around this plant. If you extract a plant from the soil and all you see are clean white roots, then it basically doesn't have a microbiome, or not a functioning one anyway. And I think this might be one of Professor White's terms, actually, that soil microbes are quiescent under most glasshouse and laboratory conditions. In other words, when we see plants with roots like this, growing in a laboratory or in a glass house. I spent a large part of my life working in the Department of Agronomy and Soil Science at the University of New England, and washed out thousands and thousands of plants that had grown in pots in the glass house, and all the roots looked like that. So I just thought that's what plant roots look like, and I think most people doing research in plant science will think that plant roots look like that. Well, when a plant root looks like that, it's very unhealthy because it doesn't have its associated microbiome, the rhizosphere microbiome. So the problem is that then most of the research that we undertake with plants that have roots that look like that is irrelevant, because they're not able to function effectively. So in a situation like that, when the roots look like that, for example, if you add nitrogen, the plants will take the nitrogen up and they will grow more. So then we form the conclusion that we need to add nitrogen to make plants grow. But if they have a healthy microbiome and we add nitrogen, they will probably grow less. So we want our plant roots to look like this, and I mentioned this on Monday. We want to have rhiza sheaths around the roots. We don't want to be able to see the roots at all. But what I want to talk about today is what happens when we have a whole lot of different plants with different microbiomes growing together. What actually happens in that shared microbiome, which I call the sociobiome, when those roots mingle? You know, what actually happens to the microbes, and how does that affect the growth of the plants? So as I also mentioned on Monday, this is a highly magnified view of what actually happens inside that rhiza sheath. So we have the plant, the edge of the plant root over here on the left-hand side, and we have soil particles over here on the right-hand side. And in between the, inside that rhiza sheath, we have all these fungal hyphae and basically trillions of microbes. And this is the world that Professor White will be talking about. He's going to be talking about what is actually happening in there and how the microbes move in and out of this plant root, which they do. And you can also see little droplets of liquid here, which are the exudates that are feeding this life. So what effect does root mingling of different plant species have on, I want to look at plant productivity, I want to look at immunity, and I want to look at tolerance to stress, just very, very briefly at those three things. So in this diagram here, we have a bean plant on the left-hand side and a rice plant on the right-hand side. The two different plant families there, the grass family and the legume family. And all the little dots on this diagram are microbes. So if we look at our bean plant here on the left-hand side, this is this black line across here obviously is the level of the soil, and we have roots and we have the rhizosphere. So the rhizosphere microbiome is quite a distinct microbiome that we can often characterize plants by that microbiome. But then they also have a phyllosphere microbiome, so that's everything that's above ground is the phyllosphere. The leaves are phyllodes, which I guess that's where the word phyllosphere comes from, but the stems, the flowers, the fruits, there's going to be microbes all over everything that's above ground, and that's called the phyllosphere. And then the third main group is going to be the endosphere. So there are microbes inside the stems, inside the leaves, inside the flowers, the fruits, the seeds. In fact, there are more microbial cells in plant leaves than there are plant cells, or there are more microbial cells within the plant as a whole than there are plant cells. In the same way that in the human body, we have more microbial cells in our bodies than we have human cells. So what can happen with all of these microbes can actually move around. So microbes from the rhizosphere can move into plant roots, and then they are in the endosphere, and they can move up into the stems and into the leaves, the flowers and the fruits, and they can actually end up in the seeds, which is what we call the core microbiome, and they will then be there for the next generation, which is what we call vertical transmission. So if a plant was finding itself in a very difficult situation, like a polluted soil, you know, with heavy metal contamination, or in a saline soil, or even under difficult environmental conditions like a drought, for example, It will often take microbes from the soil that it otherwise wouldn't have taken, and it will use the genetic material from those microbes to help it deal with that stress. And if it's significant stress, it will actually incorporate those microbes, I'm talking about living microbes now, into the seeds. And then the next generation of plants will actually be more tolerant to that stress. So let's say it was contaminated soil, some kind of heavy metal contamination, for example, and we had an annual plant growing there. Every generation of that plant will actually be more tolerant to that stress than the previous one, not for genetic reasons, but for microbiome reasons. So oftentimes when we think it's adapting genetically, it's actually adapting microbially. And that's a very, very important point to take note of, because if we look at it the other way and we say, well, how are our cultivars selected these days? We select them in a monoculture situation with lots and lots of nitrogen as a general rule. So in fact, we're adapting them to very high nitrogen soils, and we're adapting them to very low levels of microbial activity. And then we wonder why they don't survive if we don't provide all that fertilizer. But that's another story. I want to talk about what happens when we have two very different kinds of plants growing together. Can they actually share the genetics that is inherent in that microbiome, in that rhizosphere microbiome? And the answer is yes, they can, and they do. So if we just look at productivity, to give you an example of productivity increases, we know from the research that if plants have a similar microbiome, so if you had, we'll use ryegrass as an example again, we have two ryegrass plants growing together or a whole field full of ryegrass, they actually have a negative feedback. This has a negative feedback effect on plant productivity, having all the same kind of plants growing together. And that's because they become very competitive. And they sense that every other plant is the same as them. So they're competing with each other. And this has been shown in research. If they have dissimilar microbiomes, so if we had chicory and plantain and red clover and timothy or something growing together, then we have a positive feedback. So we have positive feedbacks on plant productivity coming through the microbiome if they are dissimilar. And that again has been shown scientifically to be the case. So the ideal situation, and I mentioned this on Monday, is to actually have 4 functional groups. So we try and get plants that are as different from each other as we can, while at the same time having something that's going to be productive for agriculture. So in a grazing situation, like a dairy, for example, or a beef unit, we would have all sheep. Grasses are obviously going to have to be a part of that. Legumes will be a part of that. We want some short herbs and some tall herbs. And this would be the same if we were putting in a multi-species cover. To improve soil health, we would want to have 4 functional groups in there as well. So the ideal is 4 functional groups, as different as you can make them. And then if you could have multiple species within those groups, that's even better. So just to give you an example now, in this experiment, this is Irish research, and this is Thomas Maloney's from his PhD thesis, I think. He wasn't actually looking at the microbiome, but I just want to show you what happens when you do put your 4 functional groups together. So mix 2, which is over here on the right-hand side, the blue bars, is actually a mix of ryegrass, timothy, red clover, plantain and chicory. So you have your 4 functional groups, and there's 4 different rates of nitrogen here, 0, 120, 240 and 360. 360 kilograms per hectare per year of nitrogen is equivalent to 783 kilograms of urea, so massive amounts of nitrogen. Over here on the left-hand side, we have a monoculture of perennial ryegrass, and you'll see that when we go from 0 to 120 to 240 to 360 kilograms of nitrogen per hectare, ryegrass continues to respond. So it actually has a very high requirement for N and doesn't like it when there isn't any. But what's interesting about this figure, I'm not going to talk about mix 2, is just a less diverse mix than, mix 1 is less diverse than mix 2. It actually only has 2 functional groups in it, and this one has 4 functional groups. But when we look at this highest rate, like perennial ryegrass with 360 kilos per hectare of N, I mean, that's insane, but that's 780 kilos of urea per hectare per year. It still wasn't even able to yield what or it's equivalent yield. This one is equivalent yield to a mix with 4 functional groups with no fertilizer. So if we want to cut out if we want to cut out fertilizer, we just need to go to 4 functional groups, is what I'm saying. So productivity is very much related to diversity. Drought tolerance is something I've spoken about quite often before. What we now know, and the research shows, was something that I always suspected, but we now know that in diverse communities, the plants will respond to drought by recruiting microbes from the microbiomes of dissimilar plants. They cannot do this when they are in a monoculture, because if you have a monoculture, so the example I'm going to give you is triticale. So all there is triticale plants. There's no point in one plant recruiting anything from the microbiome of the plant next to it, because they've all got the same microbiomes. So they're not able to take extra genetic material from the microbiome. When you have lots of different kinds of plants growing together, they can recruit those microbes, and we find that the endophyte content, if we look at the microbes forming the endophytes of plants that are drought stressed, the endophytes in those plants are very different to when the plant has adequate water. So I have shown this example before, but it's one of my favourites because it was just so clear. So here we have a triticale monoculture versus a multi-species mix. That was triticale plus oats, tillage radish, sunflower, field peas, faba beans, chickpeas, proso millet and foxtail millet. So we have our 4 functional groups. In a drought situation, the triticale monoculture has failed. Even though the plants are fairly widely spaced, there is nowhere for them to get any extra genetic material because their neighbours are all the same. And over here we have our cocktail crop in the corner, and our mixed species, and it's the same soil, the same rainfall, everything exactly the same, but we have plant diversity. And there is no sign of any moisture stress whatsoever in this diverse mix. So where we have plants able to share their microbiome, they can actually take up extra genetic material, and you'll find that the endophytes, which Professor White's going to be talking about endophytes to you in the next talk, very fascinating, because he's actually going to show you videos of how these microbes move in and out of plant roots. And another example I like, this one is from the Western Districts of Victoria in Australia. Again, in a severe drought situation, and here we have monoculture wheat that has failed on both sides of this green line here. And where the wheat, so the wheat has been sown right across here just as all in one pass. And here was a fence that had just been taken out. And in previous years, underneath this fence was a mix of weeds, basically. Weeds that grew at any time of the year whenever it rained, and the farmer never bothered about the weeds, just left them there. They've never been sprayed out. And now that he's taken the fence out and sown wheat right across the whole paddock, you can see what a huge difference to the soil microbiome, the fact that there was a diversity of plants. And this is the sort of thing that when farmers now, if he was to cover crop this now and start putting in a diverse mix of summer active plants in between, because we grow wheat in wintertime here, and then in summer this will just be left bare, whereas under the fence line the weeds were growing. If he was to use a summer multi-species summer mix in this every summer, he would bring the level of biological activity in this soil up to this level, and drought would not be such an issue for him. So these positive changes that we see in the soil microbiome when we have a diversity of plants, they build soil carbon, as I spoke about on Monday. They improve soil structure, as I spoke about on Monday. They increase the nutritional value of plants, and they also confer pest and disease resistance, which is something I haven't really spoken about I didn't really talk about this on Monday. So let's just talk about disease resistance, because this is something I think it's very easy for us to understand about. Well, I find it easy anyway, about the fact that, the nutritional value of the plants is going to be higher because of the diversity and, the fact that it's easy for them to actually swap genetic material, I suppose, really by exchanging microbes is what they're doing. So let's just take a look at a really classic example of disease resistance. So most of our agricultural landscapes, and I'm sure you'll all agree, wouldn't matter where in the world, well, in the Western world anyway, that we went, we would find that there's monocultures of row crops, there's monocultures of vegetables, there's monocultures of fruit and nut trees. If any of you have been to California in recent times, it is just, you'll just drive for miles through walnut trees and almond trees, and it's a very monotonous landscape because it's just going to be miles and miles of walnuts and then miles and miles of almonds or pecans or whatever, just no diversity at all. And then many home gardens are also lacking in diversity, which is a shame. So a case study on plant disease. So citrus greening, also known as HLB, is a bacterial disease that has devastated the citrus industry in Florida and the United States. So citrus production in Florida is a $9 billion industry, and it's been a recipe for disaster for some time. There's 500,000 acres of citrus trees. It's all monoculture. It's a very light sandy soil. They use high analysis fertilizers. They use cocktails of poisons. The poisons are unbelievable, would be enough to turn you off oranges forever. And there is no plant diversity, or there has been no plant diversity up until now. So it was waiting. Something was going to happen, and it did. So the Asian citrus psyllid, which is a little sap-sucking insect, I have to be careful how I say that, carries a bacterium. And so when the psyllid sucks on the sap of the citrus trees, that's like Peter picked a pickled pepper, they transfer the bacterium from one tree to the other. So this is our little psyllid here, sapping this. No, I didn't get it right, did I? Sucking the sap. I do have to be careful about that. And so this guy is actually not well, this is the vector, right? So he has a bacterium. It's like if a mosquito bites you and you get malaria, I suppose. And this is what our citrus trees look like when they have citrus greening. So the leaves go yellow, but the oranges stay green. So the oranges never go orange, and that's why it's called citrus greening. So what's been tried? $9 billion, a lot of money to go down the drain. The EPA, the Environmental Protection Agency, actually allowed or encouraged, unbelievably, that an antibiotic by the name of oxytetracycline, which is an antibiotic that's used in human use, was applied to over 480,000 acres. The whole area was sprayed in 2016, 2017, 2018. I got the impression from reading about this that you didn't get any choice. They just came in and sprayed everything, whether you were organic or not, you got sprayed with antibiotic. They've also tried injecting trees with insecticides to try and kill the psyllid, even though it's a bacterial infection, to try and stop the vector, and genetic engineering, in other words, trying to breed citrus trees that were resilient to HLB. But the problem wasn't that they weren't using enough chemicals or whatever. Like that, they have 100% of trees are now infected. All of the chemical treatments have failed. The antibiotics failed. The insecticides failed. And the multi-billion-dollar Florida citrus industry has actually been totally devastated by this disease. But the problem is not lack of chemicals. It's just a lack of plant diversity. I mean, really, it is so simple. So one citrus grower tried plant diversity, and I guess everybody probably laughed at him. Like, what difference could that make? Is what they probably said. His name is Ed James. He's now quite well known in Florida because he's leading the way, and he's demonstrating that plant diversity can reverse citrus greening. So what did he do? So here are his orange trees. He has planted multi-species covers in the interrows. It's not really that hard, is it? So he's got a 9-way mix. He's holding a sun hemp plant upside down there in that photograph. So this is sun hemp, the one with the yellow flowers, which is a warm season legume. but there's a mix of 9 other things in there, and he's got his 4 functional groups. And he does plant, I'm pretty sure, two multi-species mixes a year. So there'll be a summer mix and a winter mix. It's another photo of just when the sun hemp's in flower. So it's basically wall to wall. He's covered all the ground. And that is multi-species, even though it just looks like sun hemp there at the moment. So what do the neighbours say? Looks terrible, looks untidy. He's the only person in Florida that's got. So his citrus trees still have HLB. Now this is what's really interesting. If you test the leaves, they still have the bacteria, but they are highly productive and they're carrying on just as if, you know, they're perfectly normal. The leaves aren't yellow. The oranges are going orange. So what's going on here? What's going on here is that we have basically root mingling in the soil sociobiome. So we have all of these plants that are very, very different genetically to citrus trees, obviously, and they have different microbiomes and they have different genetics in their microbiome. And because the citrus roots go right in under here and these roots go right in under there, I'll show you a diagram of that in a minute, there is root mingling in the soil sociobiome, and they are swapping genetics, and the genes that are, the microbes that are associated with these plants have genes that are able to withstand the HLB, even though these plants still have that bacteria in them. So in these diverse plant communities, the trees can remain productive even when infected with HLB. And so now the ecologists have come along and are actually conducting research into what Ed is doing, which is great, because oftentimes what we see is that the scientific community just turns its back and says, no, that can't happen, that's impossible, and we don't want to research that because, you know, a farmer discovered that, so we don't want to know about it. Unfortunately, that's something that we hear around the world, is a story that's heard too often. But the scientists have finally got behind this, and they're now conducting research into what Ed's doing, and they're finding that plant diversity actually does support, well, we know it does, supports microbial diversity, and it is actually improving tree health. So this is a publication that came out in February this year, February 2020. So here we have our citrus trees, and you can see that some of the roots go right across through the interrow here, and then we've got our diverse cover crop in the middle, and we've got root mingling here in the soil sociobiome. And that root mingling is sufficient for genetic material. In actual fact, what's happening is that microbes from these plants are moving up as endophytes into these plants because these plants need them, and they're sending out the signals that they need them. So there's cooperation going on here because they're different. So plants will cooperate when they're different enough. If they're all the same, they don't want to cooperate, they will compete. So plant diversity restores topsoil. I talked about that on Monday. Replaces fertiliser. I also talked about that on Monday. It renders insecticide obsolete, as the citrus greening example shows. Makes fungicide redundant. I didn't talk about that, but you have to take my word for it. There's plenty of research on that. Supports beneficial insects. Now, this is really, really important. As you can imagine, if you think back to our multi-species covers and our multi-species interrows in horticulture, we're obviously going to have lots more nectar and pollen for beneficial insects, our pollinators and our predatory insects as well. Displaces weeds. There's no room for weeds if you cover everything with plants. And remember, if you make the plants very different, if they're all very different from each other, they're going to be cooperating, not competing. And it improves landscape function. And what I mean by that is that if you have well-structured soil, when you get heavy rain, it's going to infiltrate quickly and it's not going to run off so quickly to create flood situations. And then in a dry time, you're going to, what we're noticing in Australia is that if we have deep soil carbon, in other words, if we have farmers using diversity to deepen their soils and get soil carbon sequestered below 30 centimetres, it's starting to improve the subsoil, and especially if there is clay in the subsoil, that clay will be able to be much better structured and will actually hold water that plants can then use in a drought. Whereas if that clay has just become compacted as it will if the plant roots. So if I thought of an Irish situation where you're growing something like ryegrass, which is known to have very short roots, and maybe some white clover, which is also known to have very short roots, and using urea fertilizer, for example, which shortens the roots even more, then the subsoil is going to not function. It's going to be compacted. It's not going to enable water to flow properly, and it's just going to affect the functioning of the whole landscape. So it is very important for a whole lot of reasons that we have plant diversity. And finally, how does this apply to vegetable production and to home gardens? I mean, if you're sitting there thinking, well, I'm not a farmer and, you know, how does that apply to me? Well, it does apply to you if you grow your own veggies. So the rules of the kitchen are for home gardens, no bare soil ever. Don't just go out and dig up all your soil and leave it bare. It's fine to dig it if you want to, but put something straight in it because bare soil always goes backwards. Replace any NPK fertilisers that you are going to use with biostimulants, and there's lots of great biostimulants around that you can buy or you can make your own. You can have a worm farm and use your own vermi liquid or something like that. But read up about that, find out about biostimulants. They're not replacements for fertilisers. They are things that will stimulate biology. So if you were to look at something like compost extract and you go, oh, it's hardly got any nitrogen, it's hardly got any phosphorus, how can it work? Well, the whole point is you don't want it with nitrogen and phosphorus in it. You just want something that's high in autoinducers that's going to stimulate biology. And plant more flowers. I think that would be the mantra, whether it's a cover crop, whether it's a pasture, whether it's an orchard, or whether it's your home garden and your vegetable garden, plant more flowers because they're going to support beneficial microbes as well as beneficial insects. And we want to link those plant microbiomes. Remember, you want dissimilar plants from different plant families because they will cooperate and not compete, and they look pretty. So you might have like raised garden beds with just stick some flowers in between your veggies or something like this. Alyssum, by the way, is a brassica. It's in the same family as cabbages and cauliflowers and stuff, and it's edible. But I wish you luck because I've tried it several times and I didn't think it tasted all that good, but it won't kill you. And the bees love it. There's always bees around Alyssum. And even for your trees, don't have bare ground around your trees and don't have grasses around your trees. Grasses are very competitive with trees. Plant pretty flowers and have some herbs and things in there. It's going to be better for the tree and better for the insects, and in fact better for the entire landscape. So that's all the information that I wanted to provide today, and I'm ready for questions, John.
Fantastic, Christine. It was really terrific. We were just saying here, I think we've all learned a lot, and I certainly have. And I'm going to go straight to questions. Our first one is from Jonathan F. And he asked the question, he said, to fix imbalanced soils, should we add mineral amendments or focus on building soil biology to access minerals?
Well, I would probably say that neither, really, because if you increase plant diversity, I think when he says, should we build soil biology, he probably means, should I add microbes? What you need to do is support the microbes, and you want to support lots of different kinds of microbes. So you want lots of different kinds of plants. So there's very few situations where you cannot think of some way that you could increase diversity. If he increases plant diversity, he'll improve soil function and plant function.
Okay.
But you don't need to add the minerals, no.
Okay, that leads nicely to a question from somebody, I'm not sure it's their name, they're called Sheep Geek, so it may be a pseudonym. And Sheep Geek asks, Christine, are there any beneficial microbes shared between the soil, plant and grazing animals?
Yes, I would say yes, and we've now know that with the human microbiome as well, that some microbes that are the endophytes, which Professor White's going to be speaking about, if you were to eat something like some fresh parsley or fresh coriander or something like that that had been grown in biologically active soil, and had lots of beneficial endophytes in it, in other words, microbes actually living within the plant, it's now been shown that they can get through, like in the human digestive system, they can get through into our gut microbiome and can improve the diversity of our gut microbiome. So then with the animal that was grazing, it would depend whether it was a monogastric like a horse or a pig, or whether it was a ruminant animal. And if it was a ruminant like a cow or a sheep or something, what we know is that plants that have lots of secondary plant compounds, so things like your plantain and chicory, those kinds of forbs that have high levels of secondary plant compounds in them, actually stimulate rumen function and improve feed conversion efficiency. So they're very beneficial to animals compared to something like just like a grass, a monoculture of grass that is very low in secondary plant compounds. So the plant diversity is very important for animal digestion and animal health and the conversion of feed to whatever it is, meat, milk or wool or something.
And has anybody looked at it from the point of view of animals? Do they thrive on a varied diet? We humans, for example, we tend to obviously prefer variety in our diet. Have we looked at whether animals also thrive better when their diet is diverse? Because, for example, obviously there's been a big move towards monocultural rye grasses, which grow very well and so on. But do we know anything about how animals feel about this?
Yes, we do. We know that dairy farmers will tell you that if they open the gate and the cows go in and it's just ryegrass, they will just stand there and bellow, like as if, is there nothing else but ryegrass? And that farmers who are starting to, let's just say, use the dairy example, they're starting to transition to having multi-species pastures on their dairy farm. They say the cows know which paddocks is multi-species, and they are breaking down the fences to go back. So they'll graze them there, then take them in and milk them, and then take them to another paddock, which might be ryegrass, and then the next day they come back and they've left the ryegrass paddock and they've broken the fences and they've actually gone back. So I'd say they definitely like the multi-species, and the milk production goes up every time they graze the multi-species pasture. There are so many benefits, so many productivity benefits, and so many animal health benefits. The other thing I hear from around the world, again, just talking about dairy farmers, is that at one time they were calling the vet like almost every other day for some kind of metabolic disorder in cows, because when cows are just on ryegrass, they get laminitis, which is like their feet get really sore, they get mastitis, they get liver dysfunction. There's all kinds of metabolic issues with cows on ryegrass. And then as soon as farmers transition to multi-species swards, they say, we've even forgotten the vet's phone number. I mean, the only reason they might call the vet is if there's a problem with calving or something like that, or to do routine things like pregnancy testing or those kinds of things. But definitely the metabolic issues just disappear. And I hear that time and time again, that we just don't have to call the vet anymore, you know, for the sorts of things we used to before. So it's very beneficial for animal health.
Okay,
in fact, I would go as far as to say it's an animal welfare issue to have them on just ryegrass. I think that's an animal welfare issue. I think it's cruel for them to be just on ryegrass. Okay. I think they should be given a multi-species mix.
If I can add just a rider to that question, it's not uncommon for farmers to be encouraged to burn off ryegrass using a herbicide after a number of years and basically reseed from scratch. What impact does that have on the underlying microbiome?
Well, that has an impact on a lot of things, John. One thing that happens is that farmers will use glyphosate to burn off a paddock and then say, well, we don't want to waste that grass that we've just applied the herbicide to, so they'll put the cows in and graze it. And that's very common, that the cows are actually grazing that glyphosate, and then it will end up in the milk. And the other thing is that it's translocated to the soil, and it does affect the soil microbiome. So glyphosate has been shown to affect the uptake of important trace elements and minerals from the soil. So it is very detrimental. So if I was myself given a choice between, if I was a farmer and I wanted to go from a ryegrass paddock to a multi-species sward, I would actually just get in and cultivate it and get my multi-species in there. I would never use glyphosate myself. would never use it. And I think it will be banned eventually. So people have to get used to the idea that it's not going to be around. And also that consumers are going to not tolerate having glyphosate in their food because, I mean, just about every person in Europe, and I'm sure the same would be in Ireland, but I don't know about Ireland, but like if you go to places like France, Germany, whatever, every single person that has been tested has glyphosate residues in their urine and in their blood, and all of the women who've been tested that have babies have glyphosate residues in their breast milk, every single one. So at some stage, glyphosate is going to be banned. The people need to think about what's going to happen when it is banned. Get ready for it. And you don't need it. It's not essential because the problem is that we've got this thing about, oh, you can't cultivate the soil, you can't touch the soil. Spraying of poisons into the environment is much worse than getting in. Just cultivate it and get your multi-species mix in there and just don't leave it bare. The big problem with cultivating soil is leaving it bare. If you plant something straight away, then that's fine.
Okay. I'll move on, Christine. I have a question from Frank Gruneweg in Montana. And Frank asks, in an annual cropping production system with 5 months of cold winter, what can be applied in furrow to kickstart biology?
Well, when everything is frozen in wintertime, it doesn't really matter because it's been adapted to that, for millions of years anyway. And when it warms up in spring, at the same time as plants are growing, then the microbes are going to kick in. And I presume he means, he's not talking about the middle of winter. He's talking about when he plants something in spring, or he might plant a rye cover crop in fall, and it will just sit there over winter, and then it will come to life in spring because rye is very cold tolerant. So the kinds of things that he could put in furrow would be a biostimulant like a vermi-liquid or a compost extract, or there are several products that, like commercially, I won't name them, but there are several commercial biostimulants that are very, very good. You can make your own or you can buy a commercial one. It would be best if you could put it on the seed because when the seed is germinating, when the plant is germinating, and you want it to form a relationship with the soil microbiome immediately. Like the best relationship that it can form, it will be like a newborn baby. If it gets good microbes from its mother, then it's set for, and then has breast milk that doesn't have glyphosate in it, hopefully. You're setting that baby up, for a really healthy life. And so when the seeds are germinating in the soil, we actually want them to have lots of beneficial biology around them. So all you need is a biostimulant that's high in auto-inducers and a vermi-liquid or a compost extract, particularly from fermented compost. I don't mean compost tea, totally different thing to compost tea, or fermented seaweed or any fermentation product. I think Chris Trump is going to be speaking about Korean natural farming. Is that tomorrow? So yes. Korean natural farming is actually a fermentation process, and that would be perfectly fine as well. On the seed and in furrow would be great.
Okay. Moving on, Stephen Z asks the question, are microbes shared between plants via AMF networks? And do AMF and EMF networks interconnect?
Yes, AMF and EMF do interconnect. So what he's talking about is arbuscular mycorrhizal fungi and endomycorrhiza and ectomycorrhizal fungi, and yes, they do. So we have your trees are going to have a lot of ectomycorrhizal fungi, and then if you had a cover crop in between, like with the citrus greening, the cover crop plants would all have endomycorrhiza, which are your AMF. So those mycorrhizal highways and the common mycorrhizal network are very important for messaging, for sending signals, and also microbes can move along those. And that is one way that microbes can move around. But also if we have root mingling, this is why it's very important for roots to actually mingle in the soil microbiome, which is why I my subtitle was root mingling in the soil sociobiome. If plants' roots are close together, then they can interchange the genetic material. The genetic material is actually microbes. Plants are actually swapping microbes because the genes are in the microbes.
Yeah. And there is a question here from Jack Healy. Now, I think you've actually answered it already, so I'm going to summarize it briefly. Jack asks, do routine reseeding of pasture, does it destroy the plant microbiome and therefore reducing drought tolerance? Now, I think we've pretty much covered that one, and I think we can say to Jack that that is the case. Is that right?
Depends what you mean by routine reseeding. If you mean spraying with glyphosate and then just putting in a monoculture and using lots of fertilizer, the answer is yes.
Yeah.
But if you were planting a multi-species sward and you were using a biostimulant, then the answer is no, because if you're putting when you reseed, if you're putting multi-species in and you're establishing good microbiome around the roots of those plants, then you will improve drought tolerance, because plant diversity will improve drought tolerance.
Okay, yeah, I suspect that Jack is talking about clear out and reseed. So another question, Christine, for you from Sam Dean, and he asked the question, could a short or tall herb also be a legume within the 4 functional groups?
No. I'm sorry.
Okay, well, that's a quick
You have to be creative there. Yeah, so you can have grasses and legumes, but you must have tall, non-leguminous herbs and short, non-leguminous herbs. You must get away from just grasses and legumes.
Okay,
you have to have some other things in there.
Okay, and now from Sheila Cook. Sheila asked the question, she said, let's say you have a dead soil, what would be the quickest ways to rebuild the microbiome?
The quickest way is through plant diversity, and if the soil was really dead and you wanted to get it up to speed as fast as possible, a large diversity or wide diversity of annuals will do it faster than a diversity of perennials. So you would probably, Ed James with the citrus greening, he put in, like, I think it was a 9-way mix of annuals, and he does that twice a year, and he really just absolutely blitzed, the microbiome just improved out of sight on his farm. He has people from all over the world now coming to see what he's done, and all he was doing was just putting annuals in twice a year. So I think Sheila's in England, so whether you can do that twice a year, I'm not really sure, but at least a good mix of annuals for a while, and then you'll notice that soil structure will improve hugely. The smell of the soil will improve, the colour will improve, and when it gets to the point of being much, much better than it was where it started, you could say, okay, maybe it's ready for perennials now, because once you put a perennial mix in there, if you manage it appropriately, that could be forever. Once you put a perennial mix in there, you should never have to touch it again. So this whole issue of, someone raised a question before about reseeding pastures. Reseeding pastures will become unnecessary when you have a diverse perennial mix that's appropriately managed.
Okay, so...
And by appropriate management, I mean appropriate grazing, and you've had several people like Greg Judy talking about grazing.
So we should be aiming for continuity rather than the big build-up and then the blitz and start again. I'll move on, if I may. Debbie Wilkins asked the question. She said, if seeds have dressings, is it going to kill the seed biome? Or is adding biological seed dressing helpful?
Usually when people mean a dressing, like when you buy dressed seed, it usually means it's got fungicide and insecticide on it, or neonicotinoids, something on it.
Yeah.
That's what dressed seed usually means.
Yeah.
But now some of the seed companies are actually selling seed with a biostimulant on the seed. So the choices are, number one, do not buy seed that has poisons on it, because yes, it does affect the microbiome. So your other 2 options are to buy what's called bare seed or raw seed, and you may have to buy that from another farmer, or you may need to tell the seed company that you will not buy it with poisons on it. And that's what's happening here in Australia, is that farmers are now refusing to buy the seed that's got the insecticide, the fungicide on it. So the seed companies have to provide bare seed for them. Or the seed company's saying, okay, I'll sell you seed that's actually got a biostimulant on it, so that you now don't have to apply the biostimulant. And that's becoming the big trend now, especially with multi-species mixes, for the companies that are selling multi-species seed mixes to actually sell them with a biostimulant, which is not a fungicide or an insecticide, but actually a positive, helpful thing for the biology. That will become the way of the future.
Great.
Is mix seeds and coming with a biostimulant already on them, so the farmer doesn't have to do that.
Okay, one here from Tim Cammon. And Tim asks, how do we cheaply convert our ryegrass farming landscape to multi-species swards without having to reseed the whole country? I'm sure a lot of farmers tuning in have been very, if you like, stimulated by your suggestion that what we need to replace monotony in our landscapes with is diversity, but can we do it cheaply and effectively without reseeding the whole country?
On Monday, I spoke about a machine that was developed in Australia that's a kind of a strip tillage machine. The farmer that developed that was the first Australian farmer to be awarded carbon credits under our government regulated scheme that's part of the Paris Agreement for building soil carbon. And so it strip tills 17% of the soil. So the rest, what does that leave? 83%, is that right? So 83% of it isn't cultivated. So if you had ryegrass, you could go in and strip till it with a machine. Like I'm sure people can build something very similar and plant your multi-species into that area. And then over time, you might come back and you might do that again, but you're never actually totally destroying what's there. You're leaving most of what's there and you're putting something extra into it and you're using a biostimulant with it as well. Stop using your high analysis fertilizers. Start putting diversity in, and it's a way that you can do it without, creating massive destruction or having to use poisons. It's just a gradual transition, if you like, and it's very effective.
Can I inject a question as an innocent person myself? Why do you think farmers are being pushed towards such a adversarial relationship with their own land, given that it's clearly in the long run it's not working? Who's pushing them?
I don't think it was intentional, and having come from a research background myself and having worked in a Department of Agronomy and Soil Science, as I said, all the plant roots that I ever saw as a young researcher, which is quite some time ago now, but they were always white, they were always clean, because we grew plants in the glass house in soil that had been sitting in a bunker for probably 6 months or 12 months or something, had no life in it, and oftentimes, soil is sterilised before it's put into pots, or it will, at the very least, it'll be homogenised. So if you had a 200 pot experiment, for example, and you wanted to compare different rates of fertiliser, you're going to homogenise the soil so that every one of those 200 pots has got soil that's pretty much the same, and sometimes it'll be sterilised. So it's dead soil, and you put the plants in and they don't act as a holobiont, they don't have those rhiza sheaths on the roots, they have clean white roots. And so they're going to react very, very differently to how a plant would react naturally. So we came to all the wrong conclusions. We came to the conclusion that we needed to add fertiliser to make plants to grow. Whereas in a diverse situation, like you go to a diverse meadow, okay, and you've got lots of flowers and things in there, we don't need to add fertiliser. In fact, if you add fertiliser, you probably lose a lot of the things that are in there. So we came to the wrong conclusions, but I don't think it was intentional. And the problem is we've gone further and further down that track, and now there's a lot of egos invested in maintaining the status quo, because to admit that we did the wrong thing, it's very hard for some people.
And presumably as well, there's a lot of money, if you like, in selling these inputs to farmers, and that's obviously a challenge to, if you like, counterbalance some very strong commercial interests in continuing for farmers to use these.
Yeah.
Yep.
Yeah.
That's very true, John. In fact, 98% of the gross revenue that comes from agriculture goes to input providers. So that's not just for seed and fertiliser, but it's also for tractors, and it's also for accountants and bank loans and all of the ancillary industries that go with agriculture. 98% of the gross revenue goes to those.
Yeah.
The farmer gets 2%.
Yeah, so it's a system that's working extremely well for some people, but not necessarily for the actual primary producer, for the farmers themselves.
Absolutely.
And also, I'm assuming most farmers care deeply about their land. They probably inherited from their parents, and they would like to leave it to their kids, perhaps, and presumably they'd like to do it in as good a condition as they inherited.
I agree, and I would like to see 98% of the revenue go back to farmers rather than going to input providers, and that means that they need to start looking at these natural processes, like how do plants work cooperatively together, how can we do away with the fertilisers, with the insecticides, with the fungicides, with all of those things. There are so many natural alternatives, and that's what Regen Ag is really about, and it's about networking, it's about farmers sharing that information, helping each other. And that's why, like the Danu farming group that you have in Ireland is about helping farmers transition away from conventional ag to more biology-friendly ag. And also, what's happening with what Sean and Kevin are doing there, and what you're involved with as well, is, it's a movement that's building, and it's fantastic to see. I think it's very exciting and it's very, very positive for the future.
Fantastic. Now, the clock is against us, Christine, so I'm going to come to a question from Declan, who asks, if a diverse cover mix benefits trees, can the same be said vice versa in an agroforestry situation?
So that a diversity of trees would be a good thing to have.
If a diverse cover mix benefits trees, could it be said could the same be said in an agroforestry situation?
You mean a diversity of trees would be of benefit?
I believe so. I believe that's what he means, yes.
Yes. Well, either way, a diversity of trees would be of benefit, and also in an agroforestry situation, a diverse pasture would be very beneficial to the trees.
Yeah. Okay.
A diverse pasture under the trees would be very beneficial.
Okay, and I'm going to finish, if I may, with a very short question from Sandy Roberts, and they ask, how do you get this diversity in lawns and still keep the aesthetics?
Well, the only good place for a grass is in a lawn, as far as I'm concerned. Grasses make good lawns. They're not very good for anything else. So I don't have any problems with people having a grass lawn. I think that looks fine, as long as they don't think that they can have a dairy pasture that looks like a lawn.
Fantastic. Okay, so Sean, do you want to come in? Yeah.
Thanks very much. Again, another inspiring talk. I have two more questions I wanted to ask you. One is from the horticulture side. Rachel Hardiman says, does the use of multiple species in a vegetable growing context lessen the importance of crop rotations?
Yes.
Simple as that.
Well, because yes, I mean, if you just put flowers in between your vegetables, and the flowers that you could use would be things like flax and buckwheat and cosmos. Like, it's going to depend what time of the year it is as to which flowers you would grow. But there are lots of flowers that kind of grow straight up, like delphiniums and things like that don't take up much room. And you can slip them in. And those photos I showed, like marigolds, violas are another great thing. You know, in other words, so you can never actually see the soil. You just have flowers in all the spaces, especially for plants like, say, cauliflowers and cabbages and things that tend to have a lot of space between them when you're starting out. You know, if you cover that ground around them with flowers, it's going to be great for the pollinators and the predatory insects and all those kinds of things. And it is really building the soil microbiome, and probably, provided you had diversity within your vegetable mix, you wouldn't need to worry so much about rotation.
Okay, and my final question is, was an email into me from a woman in the west of Ireland who's saying that she's a problem in her with silverweed, which is, I can't remember what silverweed is now, but is there any way that she can increase biodiversity without ploughing and to reduce the weed impact that she has a problem in a lot of her fields?
I don't have an answer to that, Sean, sorry, because I've got no idea what kind of a weed it is that you're talking about.
It's Potentilla anserina.
Oh, that's helpful. Sorry. I don't know.
You don't know, that's fine. I would imagine it's the same thing it's a perennial application of mixed species again.
I guess so. Yeah, I'm not familiar with that weed. I don't. Sometimes cultivating certain kinds of weeds, when you cultivate them, you chop the roots up and you actually end up with more, because each little bit of root, you know, grows into another plant. So without actually knowing what the plant is, I wouldn't be able to say. But for most, I would suggest that cultivating and putting in a multi-species sward would actually be a good answer.
Yeah.
Well,
but I'm not familiar with that weed, sorry.
Thank you so much. And we now know it's past 3 a.m. where you are, so you've been fantastic to stay with us with such stamina and such energy and such passion in your presentation. It was really terrific. And so I think on behalf of everybody here in Carrigan Shannon, we'd like to say thank you so much.